DHammy Presents AS1 Biology, Molecule and Cells, full revision video for CCEA. Our first topic today will be water. The elements present in largest proportions in living organisms are hydrogen, oxygen, nitrogen, phosphorus, sulfur, and carbon. Water is dipolar, and dipolar is when oxygen is slightly negative and hydrogen is slightly positive. Hydrogen bonds are also very weak bonds. They have a polar nature as the water results in formation weak hydrogen bonds between the water molecules. Hydrogen bonds are found between the oxygen and one water molecule and hydrogen atom on the other. Hydrophilic is water loving, hydrophobic is water hating. Our next topic is going to be inorganic ions. Calcium is the metal lamella of plant cell wall and bones and teeth in mammals. Iron is an essential component in hemoglobin. Magnesium gives chlorophyll. Potassium maintains electrical gradients across neurons. Nitrate is amino acids and nucleic acids in chlorophyll. Phosphate is phospholipids. Phosphate is the main component of the cell membrane. Hydrogen and carbon is important as a buffer. A buffer is a chemical or substance that resists changes to pH. Example: albumin and hydrogen carbonate. And an osmotic system is the movement of water in relation to water potential, such as high to low concentration. Our third topic is going to be carbohydrates, and it's our first major topic. Carbs are complex carbon-containing molecules formed of monomers joining together, forming polymers through polymerization. They contain carbon, hydrogen, and oxygen only, and they're split into monosaccharides, disaccharides, and polysaccharides. Monosaccharides have a general formula of CH2ON, and the types are alpha glucose and beta glucose. An isomer has the same molecular formula but a different structural formula. Alpha glucose is the basic subunit of starch and glycogen, and beta glucose is a constituent monomer of cellulose, and fructose is found in plants. Here are diagrams of alpha glucose and beta glucose. Here's a diagram of fructose. Disaccharides have a general formula of C12H22O11, and they form when two monosaccharides react together as a condensation reaction and have a 1,4-glycosidic bond. A disaccharide molecule is broken down into monosaccharide monomers in a hydrolysis reaction. Here is an example of maltose. Polysaccharides have a general formula of C6H10O5N, and they form through condensation reactions, and important polysaccharides are starch, glycogen, and cellulose. Starch is a storage polymer of alpha glucose. Amylose has 1,4-glycosidic bonds coiling, forming a spiral, and are held in place by hydrogen bonds. Amylopectin has 1,4 and 1,6-glycosidic bonds. The benefits of starch is that they're very compact. They contain a rich store of glucose in a small space. They're insoluble in water, so don't affect water relations of the cell, and they're large molecules, so they don't easily pass through the cell membrane. Here's an example of starch and amylose and amylopectin. Here's an example of glycogen, and here is an example of cellulose. Glycogen is alpha glucose monomers, 1,4 and 1,6-glycosidic bonds. Chains are branched. They're storage carbohydrate found in animals and fungus. Cells are compact and insoluble, so they don't affect the water relations, and they have more terminal end of the branches, so factor hydrolysis. Polysaccharides are well adapted for storage as they have many glucose monomers in the cell, readily hydrolyze, releasing glucose molecules. They're branched so many terminals, and they're osmotically inert. Cellulose has a role as structural made from beta glucose with 1,4-glycosidic bonds. The rotation of one beta native structure is different from alpha. Megafibril consists of hundreds of cellulose molecules, structure of immense tensile strength. They form a cell wall preventing the cell from bursting. The flipping of adjacent glucose monomers has two effects. The unbranched chains are straighter, and the hydrogen bonds forming cross linkages between adjacent chains. Here's an example of the cellulose molecule. Lipids will be our next topic. Lipids contain carbon, oxygen, and hydrogen, and they're insoluble in water, but they're soluble in solvents. There are two types: triglycerides and phospholipids. Triglycerides are formed through the combination of glycerol and free fatty acid molecules. The formation is when a condensation reaction between the OH and glycerol and COOH carboxyl group of each fatty acid, as an ester bond is formed for each condensation reaction. So one glycerol and free fatty acid gives a triglyceride or free ester bonds in a condensation reaction. Here is a diagram of this occurring. Saturated fatty acids contain carbon atoms of CC single bonds, and unsaturated fatty acids have at least one CC double bond. Monounsaturated chain only contains one CC double bond. Polyunsaturated chain contains more than one CC double bond. And triglycerides are unsaturated or oils at liquid or room temperature. Triglycerides are saturated or solids, and so are fats. Properties of fats and oils is that they release more energy than carbon fatty acids, and they only respire aerobically. Fats are important in providing the thermal insulating layer in mammals, buoyancy in marine animals, and cushioning layer to protect internal organs. Phospholipids are formed by combining a glycerol and two fatty acids along with phosphate. The head is glycerol and phosphate, so hydrophilic and they're soluble in water, whereas the tail is fatty acids and hydrophobic, so insoluble in water. Phospholipid bilayer represents the basis of membrane structure in a cell. Steroid cholesterol is hydrophobic, and steroid hormones are synthesized from, including hormones like estrogen and testosterone. Here's a diagram of a phospholipid. Proteins is going to be our next topic. Proteins function as enzymes, hormones, and oxygen transporters to form the bulk of skin, hair, feathers, nails, and cartilage. Proteins are huge 3D molecules whose monomers are the variety of different amino acids found in nature. Protein structure and function is when proteins contain carbon, hydrogen, oxygen, nitrogen, and usually sulfur. Their large polymers formed from amino acids, and the shape is determined by the sequence of amino acids. The formation of a dipeptide is when amino acids are linked together by a peptide bond involving condensation reactions. The loss of water and condensation reaction takes place between amino group and the carboxyl group. The amino group is joined together forming a dipeptide. This is an example of how a dipeptide is formed. Primary structure is a sequence of the amino acids in the polypeptide chain linked together by peptide bonds. Secondary structure is when alpha helixes or half-hitch bonds form between amino acids and bonds twist the chain of amino acids into a spiral shape and are held in an alpha helix shape by hitch bonds which help stabilize the shape. Tertiary structure is the further folding of the secondary structure and a unique 3D globular shape of a range of bonds formed between the R groups, and myoglobin is an example. Hydrogen bonds are relatively weak. Ionic bonds are between the amino and carboxyl group and they're damaged by changes in pH. The disulfide bonds are covalent bonds between the R groups and they're very strong, so important in giving strength, and hydrophobic interactions are between the R groups. Myoglobin is a globular protein found in muscle cells. The quaternary structure consists of two or more polypeptide chains, and conjugated proteins include glycoprotein, which is important in membrane structure, and hemoglobin, which consists of four polypeptide chains, each attached to an iron-rich heme group, which is essential in the transport of oxygen. Protein groups. Fibrous. Polypeptides are arranged in chains to form fibers or sheets. Parallel chains are linked by cross bridges to very strong stable molecules, and they're invariably structural in function. Collagen is an example of three identical polypeptides held by hydrogen bonds found in tendons like in muscle to bones, keratin, and nails and hair. Globular forms specific 3D shapes, hydrophobic at the center but hydrophilic at the edges, so soluble in water, and they have metabolic rules such as enzymes, and hemoglobin is also globular. Here is the comparison between the two. So if you're asked a question to compare the two, you'll be able to. Prions is going to be our next topic. Prion is derived from protein and infection, and malformed proteins called prions cause diseases such as TSE, BSE in cattle, and CJD in humans, which lose physical coordination. In humans, the memory and body control are also lost prior to death. It involves the replacement of the normal cell surface, secondary structure PRPC, and alpha helixes. PRPSC enriches beta sheets, and so presence of PRPSC causes normal PRPC to alter secondary structure. Prions are stable, being resistant to extreme temps and radiation. They are rare but fatal. Prion disease arises in three different ways: transmission through the consumption of infected food or open wound exposed to the environment, or a medical procedure involving the affected material. Inheritance, which is a gene mutation occurs during meiosis, and mutant gene codes for the synthesis of misshapen prion proteins. Sporadically, where the normal protein PRPC spontaneously transforms into the disease form PRPSC, and no evidence of inheritance or transmission, and the average age is around 65. Nucleic acids will be our next topic. The subunit of nucleic acids is a nucleotide consisting of pentose, phosphate group, and a nitrogen at its base. Three components are combined due to a condensation reaction to form a nucleotide. Phosphodiester bond is formed between the sugar and the phosphate of the next nucleotide. Nucleic acids are broken down by hydrolysis to nucleotides. DNA is two anti-parallel strands held together by hydrogen bonds between adjacent bases. Adenine only pairs of thymine, cytosine only pairs of guanine, and DNA is a double helix. RNA is a sugar ribose and they are single-stranded. They are invariably much shorter than DNA along with the base, uracil, or place and thymine. Messenger RNA carries a code from DNA and the nucleus to the ribosome and the cytoplasm where protein synthesis takes place. Transfer RNA carries amino acids in mRNA where protein synthesis takes place and it's single-folded into a cloverleaf shape. Ribosomal RNA is made in the nucleolus and it forms over half the mass of each ribosome. Here's a table of a comparison between DNA and RNA. DNA replication. DNA is a stable molecule and DNA replication is a process which replicates DNA. Step 1 is when the enzyme DNA helicase unzips two strands of DNA breaking hydrogen bonds. Original strands then become templates for the formation of two DNA molecules. Free nucleotides are linked to the template in the correct sequence. New strands joined together by the enzyme DNA polymerase, allowing each new DNA molecule to contain one original and one new DNA strand, known as semi-conservative replication. The medicine style is a device experiment in 1958 which grew bacteria in a medium in which nitrogen is supplied in a heavy non-radioactive isotope N15. DNA bacteria became heavy in generation 0. Bacteria is transferred to the normal isotope N14, but before changing and then at irregular intervals, samples were removed and DNA was extracted. The extracted DNA involved the density gradient centrifugation and the heavier molecules at the lower level in the tube, and in generation 1 it was intermediate with 50% heavy and 50% light, but then after 2 generations it was 2 bands of 50% intermediate and 50% light. Enzymes will be our next topic. Enzymes are biological catalysts that speed up metabolic reactions. Enzymes are not changed by the reaction but are reused. Activation energy is a chemical reaction that it has an energy barrier that must be overcome before a reaction takes place. Enzymes lower the activation energy to overcome the energy barrier. The reduction in activation energy enables reactions to take place at a rapid enough rate to sustain life. Enzyme action is when an enzyme acts on a substrate and the substrate fits into a special group called the active site, forming enzyme-substrate complexes. The reaction changes substrate and forms the enzyme product complex. The product no longer fits the active site and so is released. Catabolism is the breakdown of molecules and anabolism is the building up of molecules. Enzyme specificity is due to only one substrate being the exact complementary shape to the active site. Lock and key model is when the enzyme active site is the exact mass of the shape of the substrate, but it doesn't explain why heating distorts the shape of the active site, destroying ionic bonds, so the substrate won't fit in molecules similar in shape to the substrate block the normal substrate, preventing the reaction occurring. The induced fit model, however, is when the active site molds itself around the substrate, forming a precise fit. The active site changes shape, fitting the substrate, and the enzyme puts a pressure on the substrate, breaking particular bonds, lowering the activation energy. The product is then released as a different shape to the active site, and the active site returns to its pre-reaction shape. Cofactors are non-protein substances that enzymes require in order to function. Metal ions change the shape of the active site, chloride ions and phosphatic ions, or phosphatic groups. Coenzymes are non-protein organic molecules necessary for enzyme action. They're not permanently attached and important, but they are important in respiration photosynthesis. Substrate concentration affecting enzyme activity. If they keep the number of enzymes at a fixed rate but increase the number of substrates, the rate will increase as there's more substrate available to the active sites, but the number of enzymes becomes a limiting factor. If you increase the enzyme concentration, then the number of substrate will be at a fixed rate, the level of enzymes will be increased, so the enzyme activity will increase as there's more enzymes available to the substrates, and so the rate increases, but the number of substrates become the limiting factor. If you increase the temperature, then this gives both the substrate and the enzymes molecules more kinetic energy, and so they'll move faster, increasing collisions between substrate and enzymes. Mammals is optimal at 40 degrees, and above the optimum, it causes the bonds to break and causes the active site to change shape. At around 60 degrees, the change is so great that the enzymes cease to function and it has become denatured. The effect of pH and enzyme activity. Each enzyme has an optimum pH, and either side of this optimum, the change in pH reduces enzyme activity. Caused by the pH disrupting the bonds which are important in determining the protein shape. At a low pH is a high concentration of H plus ions from negatively charged R groups lose the charge, but at a high pH is a low concentration of H plus ions, so the positively charged R groups lose their charge. Enzyme inhibition is when enzyme inhibitors are substances that interfere with enzyme action. They reduce enzyme action directly or indirectly, affecting the functioning of the active site. Competitive inhibition is when the inhibitor competes with the substrate for the active site. They are similar in shape to the substrate. The quantity of the inhibitor substance is low compared to the substrate, has little or no effect. If it's high, then it does affect it. Non-competitive is when the inhibitor attaches itself to the other enzyme other than the active site, and leads to the active site changing shape and no longer being complementary in shape to the substrate. The enzyme's second site where the molecules are attached is known as the allosteric enzyme. The effect of inhibitors, in competitive, the more substrate available, likely substrate molecules find the active site and the substrate concentration is increased, the effect of the inhibitor is reduced. But non-competitive, as they're not competing for the same active site, the increase in the substrate concentration doesn't decrease the effect of the inhibitor. Immobilized enzymes. Immobilized is when confined so that they cannot move but still act on the substrate. The commercial advantages is that production takes place continuously, Product enzyme free, so purification costs are reduced. The enzyme remains separate from the reaction mixture, so reused. Enzymes such as porous stability is improved. They remain active over a greater range of pH and temperatures. Methods of immobilization are adsorption, covalent bonding, cross-linking, encapsulation, entrapment. Immobilization also reduces enzyme activity but increases enzyme stability. Here's the advantages and disadvantages, but I'd recommend using the advantages that were shown previously. Diagnostic reagents such as biosensors. Biosensors use immobilized enzymes detecting particular molecules. Reaction changes color and used in agriculture, medicine, and industry. Glucose test strips are when oxidase acts on glucose present of oxygen yielding gluconic acid and hydrogen peroxide. The glucose color change strips is the clinosticks, but glucose meters measure the level of glucose in the blood droplet. The gluconic acid produced converted to the electrical signal monitoring the blood glucose levels for diabetics. And pregnancy test strips is when enzymes are linked to antibodies check the presence hormone chorionic gonadotropin in the urine. Enzymes as biomarkers of disease. If the heart muscle is injured, then the enzyme CPK2 leaks and testing presence in blood serum confirm if the heart attack is suffered. White blood cells gather on the urinary tract, becoming infected. The race variant of esterase testing for it in urine indicates white blood cells with urinary infection. A respiratory infection is when certain white blood cells accumulate in the respiratory tract, releasing esterase and testing the sputum, allowing for early and effective treatment. As elastase hydrolyzes the structural protein elastin, which is within the lung, reducing the lung function, and treatment involves antibodies tackling infection. Enzymes as therapeutic drugs. Therapeutic drugs as inhibitors as drug inactivates the enzyme associated with the particular medical condition, not with the normal physiological activity. Our next topic will be viruses. There are two types, bacteriophage and the human immunodeficiency or HIV. Bacteriophage is when the virus attacks the bacterial cells. The DNA strand is surrounded by a capsule with a long thread of DNA packed to the head and a short tail projects. It lacks internal membranes, cytoplasm, and ribosomes. They neither grow nor reproduce without the cytoplasm of the host cell and are totally dependent on the host cell, representing the ultimate parasitism. To reproduce, they get inside the cell and hijack the metabolic processes to make new viruses. HIV causes acquired immune deficiency syndrome or AIDS. Two copies of single-stranded RNA in spherical shape. It's outside the RNA core, typical capsid, lipid bilayer with glycoprotein, delivering RNA into the host cell with the enzyme reverse transcriptase, catalyzing the synthesis of DNA from RNA. The DNA makes new viruses synthesizing from protein coats and viral RNA. In humans, HIV invades helper T cells important in the immune system, protecting against disease. HIV is passed along with sexual contact with an infected person, shared use of a hypodermic needle with an infected person, injection of contaminated blood or blood products, and the infected mother to a fetus or suckling baby. Microscopes is our next topic. There are two types of cell, prokaryotic and eukaryotic. Prokaryotic lacks a degree of compartmentalization, eukaryotic possesses, and the prokaryotic lacks a nucleus and other membrane-bound organelles. Bacteria are prokaryotic, animal, plant, and fungi cells are eukaryotic. Self-fractionization and organelle isolation technique in obtaining organelles is self-fractionization, which involves homogenization and centrifugation. The lighter optical microscope has animal cells irregular in shape. They're very homogeneous with the granular cytoplasm bounded by the cell surface membrane. Plant cells are more complex as the cell surface membrane, cytoplasm, nucleus, peripheral cell wall, and large central vacuole. Palisade cells in the palisade layer are immediately under the upper epidermis, which are rich in chloroplasts. The unit of measurement is 1 millimeter is equal to 10 to the minus 3 and 1 micrometer is equal to 10 to the minus 6. Magnification is used to calculate or estimate the size of cells and cell structures. Magnification can be calculated by measured size of the image divided by true size, and true size is measured size of the image divided by magnification. The electron microscope, the EM uses electrons rather than light to produce images using high-power microscopes with resolution, and resolution is the ability to see two adjacent points of distinct entities following magnification. Light microscope views living specimens, but EM only uses on dead material in a vacuum. Inside the EM as air molecules deflect the electron beam. Artifacts often feature often electron micrograph images, and specimen has to become very thin using specialized cutting instruments. Types of EM include the transmission electron microscope or TEM, which involves electrons passing through very thin specimen with a high resolution used for very high magnification. The scanning electron microscope or SEM involves electrons reflecting off the surface of the image as opposed to going through it, and it's useful for giving a 3D image of the surface and features can be saw. The difference is that the electron has much greater resolving power as electrons are shorter wavelength than light. Light views living organisms and living processes such as mitosis, and the interior of the EM is a vacuum, so the specimen must be dead and electron beam is not deflected. And then our next topic will be the eukaryotic animal cell. This consists of a cell membrane, cytoplasm, nucleus, and all organelles. The nucleus contains DNA chromosomes. Cells are not dividing chromosomes, so not visible. Heterochromatin is densely packed, whilst euchromatin is less densely packed. Nucleus is the control center of the cell, and DNA and chromosomes codes for synthesis of proteins in the cytoplasm. The DNA is protected from damage, but the DNA code is taken safety from nucleus to cytoplasm by the nuclear port in the nuclear envelope. Endoplasmic reticulum, or the ER, is a membrane system extending throughout the cytoplasm. 3D enclosing sites called cisternae. Ribosome organelles dotted along the outside of the membrane is the rough endoplasmic reticulum, or the RER, but all the parts don't have ribosomes attached in there, so that's the smooth endoplasmic reticulum, so the SER. The RER provides scaffolding for protein, and ER operate in distribution network for proteins. The ER is joined with nuclear envelopes facilitating RNA transport, and the SER role is at the metabolism of lipids, including the synthesis of cholesterol. Ribosomes are small organelles with small black dots in the EM micrographs found in the cytoplasm attached to the outer surface of the ER and as a site of protein and enzyme synthesis. The Golgi apparatus is a series of curved cisternae having small vesicles entering and leaving the system. Vesicles with newly synthesized protein pinch off the RER from the convex edge of the system. Vesicles with the finished protein pinched off the maturing concave phase, and the vesicles transport the protein within the cell and fuse with the cell membrane, releasing the content outside the cell. Lysosomes are very tiny vesicles containing something to be destroyed. When they fuse, forming the secondary lysosomes, which are important in phagocytes, they digest and engulf bacteria, enclosing the phagosome. They have a relatively thick membrane. Mitochondria is enclosed within a double membrane separated by an intermembrane space. It is the powerhouse of the cell where ATP synthesis during aerobic respiration takes place. Cristae tend to be more numerous and more deeply enfolded in highly active cells. Microtubules, hollow cylinders formed from tubulin and are found throughout the cytoplasm, occurring within centrioles, formed of spindle fibers, which are essential in nuclear division. In the centriole, they are in the circular formation, and they are found in also the cilia and flagella. They're part of the cytoskeleton, helping direct movement of cell organelles. The cell membrane is a boundary with the outside wall and consists of two basic components: phospholipid bilayer, forming the skeleton due to the properties of phospholipid molecules, which have hydrophobic tails away from the solution and heads mixing with the surrounding aqueous medium. Proteins are attached peripherally to the bilayer, which is extrinsic, or integrally embedded, which is intrinsic. The structure is described as a fluid mosaic model, as phospholipid molecules flow sideways, keeping the bilayer arrangement with protein floating between phospholipid. The extended phospholipid is a glycolipid containing polysaccharides bound to the membrane proteins, glycoproteins, and glycolipids. Carbohydrates are always on the outside peripheral to the phospholipid bilayer, which is the backbone of the cell surface membrane, giving the membrane the selectively permeable properties. And cholesterol increases membrane stability. It makes the membrane less fluid at high temperatures but more fluid at low temperatures. Proteins provide stability and support to help anchor the phospholipid molecules. They act as enzymes, and they're kept in an ideal place in terms of substrate availability and pH. They need replaced less often. Some membrane proteins act as adhesion heights, such as protein involved in cell recognition. Proteins transport by channels creating a hydrophilic channel, allowing polar molecules to bypass hydrophobic center of the bilayer. Gated channels controlling the positive ions into and out of the neurons during nervous conduction. Carriers, which proteins carry specific ions or molecules across the membrane. Molecules are charge grouped, so the ions have to move against the concentration gradient, and the carrier protein change shapes, carrying the substance from one side to the other. The glycolics is polysaccharides binding to the membrane protein, or phospholipids found on the outside of the cell surface membrane. Glycoproteins and glycolipids are involved in the cell-to-cell recognition, allowing similar cell types to recognize each other forming tissues. Glycoproteins act as antigens, and they act as receptor sites, important in hormone action and passage of neurotransmitters between neurons. Glycoproteins and glycolipids form hydrogen bonds, helping to stabilize the membrane. Eukaryotic plant cell and fungi. Plant cells have a nucleus, ribosomes, and a Golgi apparatus, but don't have lysosomes or centrioles. Plant cell walls, the main component is polysaccharide cellulose, laid down as microfibrils, and each microfibril has many cellulose molecules cross-linked to each other, and on the primary cell wall there's many microfibrils orientated in different and random directions. Young plant cells surrounded by the primary cell wall and is secreted by the protoplast it encloses. Secondary cell wall, when it reaches full size additional layers of cellulose, it's deposited forming the secondary cell wall. Each layer has a microfibrils oriented in the same direction, but additional layers are oriented in different directions. This lattice type arrangement gives a great strength necessary to the cell wall function, and the middle lamella is the adjacent cell linked by the middle lamella. It's made of pectin. Cellulose fibers, the secondary cell wall, is closely packed, considerable rigidity to the cell wall. The cell wall is porous and freely permeable. The function provides support, and rigid structures are very important in turgor. They restrict the outward expansion of the protoplast as the cell takes in the water. Cells are fully permeable and no part in determining what enters and leaves the cell. The plasmodesmata is the strand of cytoplasm extending between neighbouring plant cells. Chloroplast is bounded by the double membrane and closes stroma, and within the stroma is a system of membranes called thylakoids. Thylakoids are arranged in stacks called grana, and between grana membranes is less concentrated, referred to as intergrana. One or more starch grains, smaller lipid droplets produced through photosynthesis. It's a site of photosynthesis, chloroplast, and thylakoids contain chlorophyll, most densely concentrated in grana. Large vacuoles within the cytoplasm, and it's important in storage of ions, water, and it plays an important part in development of tertiary support. Vacuole cell membrane called tonoplast. Fungal cells are eukaryotic organisms that have cell walls made of glycoprotein chitin, but no cellulose and have a vacuole, but don't photosynthesize and don't have chloroplast, but they do have glycogen as a carbohydrate store for the presence of lysosomes. They possess nuclei, and cells of many species are multinucleate. Prokaryotic and magnification. The prokaryotic lacks a nucleus, and other organelles are bound by membranes, and bacteria are prokaryotes. Here's a comparison between the prokaryotic and the eukaryotic. And here is a comparison between an animal cell, a plant cell, and a fungi cell. Protein synthesis. The nucleus and the RAR. Nucleus produces rRNA, and chromosomes contain the genetic code. They determine the sequence of amino acids. The mRNA carries the code out of the nucleus for nuclear pores onto the RAR, and the ribosomes are the site of protein synthesis. Golgi body. ER vesicles containing newly synthesized protein butt off the RAR, carrying to forming phase Golgi apparatus. The Golgi primary protein is modified into the final enzyme carbohydrate or lipid, and this is added to make a glycoprotein or lipoprotein. Vesicles and lysosomes. Vesicles bud off the mature phase of the Golgi, and the secretory vesicles carry the enzyme to the cell surface membrane, which infuse with and release the enzymes. All vesicles carry glycoproteins to the surface membrane, becoming incorporated into the membrane as receptors. Lysosomes are also budded off the Golgi, and they fuse with warning organelles enclosing the membrane material, and hydrolytic enzymes digest organelle material and carry out autolysis. Magnification is a value that tells us how much larger an image is when compared to the original object. It can be calculated by size of image divided by actual size. Size of object is size of image divided by magnification, and magnification is also calculated using length of scale line measured divided by length of scale given, and this should be the one you more or less use. Meiosis. Chromosome is a long strand of DNA bound to histone. The cell cycle. Interphase is a two-gap phase, G1 and G2 separated by an S phase. Cytokinesis is when the cell divides into two daughter cells, and mitosis is the division of chromosomes. Cytokinesis is the division of cells. DNA and cell cycle. Preparation for mitosis, DNA of the cell doubles during the S phase but returns to normal for cytokinesis. Checkpoints pass before the cell progresses to the next phase. At the end of G1, growth factors are assessed, the damage is detected, and DNA repaired proceeds to the S phase, but if not, it enters resting. At the end of G2, the DNA replicated. If not, the cell cycle is stopped. The final checkpoint is that metaphase establishes the chromosomes are correctly attached to the spindle fibers before anaphase proceeds. Cancer is a mutation of the gene encoding checkpoint proteins. Controlling mechanism break down, and the cell undergoes repeated and uncontrolled division. A large mass called a tumour stops growing, encapsulated in fibrous sheath, but it doesn't travel to other locations. But if it does continue to grow, then it's malignant. Cancer treatments. DNA onset inhibited by androstenedione and cytotoxin. Two strands of DNA can't be exposed. DNA replication can't take place. Synthesis of nucleotides inhibited by 5-fluorosyl methotrexate, and formation of mitotic spindle inhibited by Taxol, and vincristine without spindle chromatids. They cannot separate. Interphase. This is a non-dividing stage when DNA is already replicated but not coiled. Prophase is when the DNA coils so each chromosome becomes stainable and it has two chromatids attached to a centromere. Metaphase is when the centrioles reach the opposite poles and the chromosomes move to the equator of the spindle, and each one becomes attached to the spindle by a centromere. Anaphase is when the centromere is dividing the movement of contraction of spindle fibers pull the sister chromatids apart towards opposite poles of the cell. Telophase is when each chromatid becomes a separate structure and now called a chromosome. The nuclear membrane forms around each group of the chromosomes. Cytokinesis in animals. Cytokinesis is when the cell divides, forming two daughter cells. The identical chromosome makeup parent cell had, but it doesn't, then the multinucleate cell is produced. Microfilaments attach inside the cell membrane and around the center. They draw the membrane inward, creating a furrow, and the membrane is pulled until the cell is divided. Cytokinesis in plant cells is when a plant cell has microtubules remaining in the central area. They guide the Golgi vesicles to the equator. Inside each vesicle, the middle lamella forms cellulose, either side forms new cell walls. Small gaps are left between vesicles, forming plasmodesmata. It is difficult to find cells undergoing mitosis as it is only occurring at a specific part of the shoot tip and division in a very small number of cells. Significance of mitosis is that daughter cells have the same genes. As the organism develops, the cells differentiate skin cells from nerve cells. Mitosis is asexual reproduction, so the cell parent divide produces cell forming offspring production of genetically identical individuals, which suits identical offspring. Meiosis. Meiosis is the process of production division, which cell division has a number of chromosomes. The process of meiosis takes place in reproductive organs with 2 divisions of 4 daughter cells. The chromosomes arrangement is different from each other and different from the parent cell. Interphase is when the chromosomes are visible and 2 centrioles are present. In prophase 1, the homologous chromosomes for bivalence. Crossing over occurs where 2 chromosomes of homologous pair align, resulting bivalent consists of 4 chromatids, and the point where the chromatids cross over is chiasmata. Metaphase 1, nuclear membrane breaks down chromosomes pairs aligned along the equator. Independent assortment is when 1 chromosome from each homologous pair enter the gamete. Anaphase 1 is when microtubules of spindle contract, pulling the chromosomes to the opposite poles of the cell. In telophase 1, chromosomes are pulled to the opposite ends, and each chromosome consisting of 2 chromatids. Cytokinesis is when 2 daughter cells are produced with each having 2 chromatids. In prophase 2, centrioles move to the opposite poles, and metaphase 2, chromosomes move to the equator and attach spindle fibers. Anaphase 2, centromeres dividing sister chromatids move towards opposite poles, and then in telophase 2, chromatids are now chromosomes as nuclear membrane forms around the chromosomes. Cytokinesis, 4 haploid daughter cells are produced, each with 2 chromosomes. Basically, mitosis occurs twice. Significance of meiosis. It's an essential part of sexual reproduction. There's genetic variation among offspring, different characteristics, and it's important for survival of species. The causes of variation are crossing over, reduction, and independent assortment. Here's a table comparing mitosis and meiosis. Cell physiology. Transport factors are the size of molecules. It's very small molecules slip between phospholipid molecules. Large particles only move in or out by osmosis. Polarity or non-polarity of substances. Non-polar move through phospholipid bilayer, and polar substances move through via proteins. Concentration of the substance either side of the membrane move from high to low concentration by diffusion. The moving into concentration gradient required and active transport is needed. Thermal diffusion is when non-polar molecules pass through the membrane unaided, and very small molecules also pass through, but water-soluble substances are unable to pass through. And diffusion is the net movement of substance from where it is in a higher concentration to where it is in a lower concentration. Factors affecting diffusion. The concentration gradient. The greater the concentration gradient, the faster diffusion. The side of the molecule, small molecules diffuse faster. Temperature, it's faster at higher temps, it's more kinetic energy. Thickness of the exchange surface, as it's very finite for rapid diffusion. And the surface area of the membrane, greater surface area, faster diffusion. Facilitated diffusion is supported by protein carriers. Protein carriers are binding sites matching specific molecules, assisting movement of molecules across membrane. There's 2 types: carrier proteins, which take in diffusion molecules and change shape and release the molecule on the other side of the membrane, and channel proteins, which are proteins with central pore enabling charged particles to pass through, gated and open and closed, allowing the control of ion movement. Active transport is when molecules are moved against the concentration gradient from low to high, and energy ATP is required. Differences compared to facilitated diffusion is that substances are moved against the concentration gradient and energy required, and mitochondria supplies the ATP. Cytosis is important in transporting large molecule tubules for carriers and the bulk of smaller molecules. Endocytosis is the movement of substances into cell. 2 types: phagocytosis and pinocytosis. Phagocytosis is the uptake of solid particles into the cell within vacuoles, and pinocytosis is the uptake of solutes and large molecules into the cell within vesicles. Endocytosis is the cell membrane invagination around the substance entering the cell from the outside. It forms a vesicle and pinches off the inside membrane. Vesicles take into the cell, and the fluid in the cell membrane allows it to reform a close gap created by cytosis. Exocytosis is the movement of substances out of the cell contents of the vesicle, released outside the cell, and it's important in secretion. Our next topic is osmosis. Osmosis is the net movement of water molecules through a partially permeable membrane from a high water potential to a lower water potential. Water potential of a solution is the tendency to take in water by osmosis from pure water across a selectively permeable membrane measured in kilopascals. Pure water has a water potential of 0. Some water molecules are not free, so they form hydration shells around the solutes, and the presence of solutes reduces the ability of water molecules to diffuse throughout the solution. A stronger solution is hypertonic, a weaker solution is hypotonic, and an equal concentration is isotonic. 0 is the highest water potential, and the presence of substances dissolved in water lowers the water potential. Solute potential is the presence of solutes and the potential of cell solution to draw in water by osmosis. So pure water has 0 solute potential, has no solutes, and the more concentrated the solution, the more negative the solute potential. Water potential is solute potential plus pressure potential, and the solute potential of the cytoplasm is the main factor in determining water potential of an animal cell. The animal cell is the water potential equaling the solute potential as no cell wall. Osmosis in plant cells. Protoplast has cell sap, and the protoplast expands until presses against the cell wall and turgor pressure. Cell wall opposes the equal magnitude wall pressure. If placed in hypotonic, then the higher water potential, so osmosis into the cell, and protoplast swells pressing against the cell wall. In isotonic, the same water potential, so osmosis to the cell membrane just start to pull away from the cell wall. And hypertonic, there is no osmosis out of the cell, so plasmolysis occurs. The story of osmosis in the plant cell. Cell takes up water, the pressure potential rises, and the protoplast presses against the cell wall and expanding. Cell rises towards zero as the cell becomes fully turgid, but the solute potential of the expanding cell is less negative, and the cell suffers a little bit of uptake of water. The cell loses water, so the membrane begins to pull away from the cell wall, and the pressure potential is zero. Incipient plasmolysis occurs, and when the cell loses water, it becomes fully plasmolyzed. Plant cells rely on turgor for support. Opposing forces of the cell membrane and cell wall create turgor for support. Large number of flaccid cells means it's wilting, and the plant cell loses too much water, the vacuole will shrink. A situation in which plasmolysis will occur is that the plant is growing in a field given too much fertilizer, or as a seed from a woodland tree carried to a salt marsh, starting germinating in the environment. Osmosis in animal cells. Nothing stops the expansion of the cell membrane until it bursts, or lysis. Lysis takes place when the animal cell is placed in a hypotonic solution, and the animal cell in the hypertonic lose water, and the shrink is called crenation. Our penultimate topic will be the ileum. A tissue is an amalgamation of cells performing the same function, whereas an organ is a body part composed of several tissues grouped together performing an overall function. An ileum is a region of the small intestine where digestion is completed and most absorption occurs. Numerous folds of the ileum increase its surface area, and folds of numerous villi and the lining cells possess microvilli, increasing the surface area for diffusion. The serosa provides a very thin, protective, and supportive lining for the alimentary canal. The muscularis externa is a circular muscle and longitudinal muscle. Contractions of the longitudinal means the pendular movement of the gut, and contractions of the circular muscle is local constrictions pushing the food along the gut by peristalsis. The submucosa is a connective tissue with many blood vessels and lymphatic vessels, crucial in transporting and absorbing food products. The muscularis mucosa is small strands of smooth muscle extended from the main muscularis mucosa to the mucosa in each villus. Contractions of the strands produce characteristic wafting movement. The mucosa is in contact with food in the gut lumen and is highly specialized, increasing the surface area as a presence of villi and microvilli. The columnar epithelium is a column-shaped cells and lines intestine. They have 3 surfaces in the cells of microvilli forming a brush border. Glucose and amino acids are transported into the epithelial cells by active transport and diffuse into the capillary network by facilitated diffusion. Lipids diffuse into the cells and then into the lacteals. The mucosa epithelium have goblet cells producing mucus, lubricating the food, and the passage is easier. Prevents damage to the intestinal wall by digestive enzymes also, but the surface remaining covered in microvilli forming a brush border. This increases the surface area of the ileum for maximum absorption. The mucosa, the villi, and crypts. The basal villi is the crypts of Lieberkuhn. They secrete intestinal juice, increasing the volume of the gut in the gut, and pan of cells produce new epithelial cells by mitosis. The antimicrobial function protects neighbouring actively dividing cells. Pinocytosis is the absorption of extracellular fluids and antibiotics generated in very small vesicles, unspecific end substances that transport the cell takes in surrounding fluids, and all solutes are present. Our final topic of this video and AS1 biology will be the leaf. The upper epidermis has no chloroplast and is not directly involved in photosynthesis. It's covered with a waxy cuticle reducing water loss and transparent, so it's not significantly impeding the passage of light. The palisade mesoville has a primary photosynthetic region occurring immediately beneath the upper epidermis, remained regularly packed in as many as possible. Many chloroplasts maximize photosynthesis. The advantage is that sugars moving to the phloem pass through freer cells as do gases, and the large surface area of most leaves facilitate gas exchange. Spongy mesoville has a cell loosely and irregularly arranged. The formation of intracellular airspaces encourages diffusion of gases for gas exchange, with fewer chloroplasts, less significant photosynthetic role, important in gas exchange. Their loose and irregularly arrangement gives a large gas exchange surface in the leaf. Xylem vessels transport water and organic ions up through the plant and into the leaves. The phloem safe tubes transport sugars away from the leaf to the plant storage region converted to starch or used in respiration. The lower epidermis is covered with a waxy cuticle, thinner than the cuticle, and the upper epidermis has less transpiration, which stomata are present, and the opening and closing is controlled by Garrett cells. Garrett cells do possess chloroplast. Turgid is so it expands and opens, the pore of the stomata facilitating gas exchange. Less turgid than the pore closes and reducing water loss. Thank you for watching this video.